objectives of this laboratory activity are: students will discover that onions are made up of cells. Students will observe onion cells under a microscope. Students will discover that their skin is made up of cells. Students will observe cheek cells under a microscope. Good day, everyone! We are Group 2 from MSc 11-3 St. Matthew. Today, we will be presenting our laboratory experiment entitled Plant and Animal Cells Microscope Laboratory Activity. In this experiment, we observed different specimens under a microscope, including onion cells, cheek cells, contaminated canal water, and ripe banana tissue. Our goal was to identify the structures of plant and animal cells and compare their similarities and differences. Before we started with our experiments, Mr. Paulo Gamera, our Biology 1 instructor, demonstrated the proper use of microscopes and the process for seeing the plant and animal cells. Onion cells are plant cells. The materials needed for this experiment include thin onion membrane, microscopic glass slide, microscopic cover slip, iodine solution or methylene blue, laboratory gown, dropper, cutter, microscope, gloves, face mask, and forceps or tweezers. For the procedure, take an onion and cut it into halves. Peel out one of the fleshy leaves. Take out the epidermal layer by breaking the epidermal layer into a small portion and using forceps to peel out the inner lining. Cut the inner lining into a small piece. Place the specimen on the glass slide and make sure it does not roll. Add methylene blue, around 1 to 2 drops, and a drop of water to the specimen. Put the cover slip and make sure that there is no air bubble in the specimen. Slowly place the cover slip on top of the specimen. Put the specimen slide under the microscope and observe. 10 times or low power first, then 40 times or high power next. Here are the onion cells under the microscope. Our group observed that onion cells are nucleated cells, have strong and distinct boundary, are rectangular shaped, nucleus is near the periphery of the cell, cells are rectangular and closely packed or brick-like, cytoplasm is stained light blue, and nucleus is stained dark blue. Cheek cells are animal cells. The materials needed for this experiment include cotton buds, microscopic glass slide, microscopic cover slip, iodine solution or methylene blue, laboratory gown, dropper, microscope, gloves, and face mask. For the procedure, take a cotton bud and collect cheek cells from the inner lining of your mouth for 1 to 2 minutes. Rub the cotton bud with cheek cells on the glass slide. Add 1 to 2 drops of methylene blue stain to the glass slide and specimen. Put the cover slip and make sure that there is no air bubble in the specimen. Slowly place the cover slip on top of the specimen. Put the specimen slide under the microscope and observe. 10 times or low power first, then 40 times or high power next. Here are the cheek cells under the microscope. Our group observed that cheek cells have nucleus at the center, no cell walls which leads to indistinct boundary, cytoplasm is stained light blue, while nucleus is stained dark blue. Contaminated water canal. The materials needed for this experiment include cotton buds, microscopic glass slide, microscopic cover slip, contaminated water, laboratory gown, dropper, microscope, gloves, and a face mask. For the procedure, take a cotton bud and dip it into the contaminated water canal. Rub the cotton bud with contaminated water on the glass slide. Add a drop of water to the glass slide and specimen. Put the cover slip and make sure that there is no air bubble in the specimen. Slowly place the cover slip on top of the specimen. Put the specimen slide under the microscope and observe. 10 times or low power first, then 40 times or high power next. The contaminated water canal under the microscope. Our group observed that contaminated water canal had tiny moving microorganisms, paramecium that is oval or slipper shaped, euglena that is elongated, tiny bacteria appearing as dots or short rods, small floating particles, debris, or sediments, and organic matter. Ripe banana. The materials needed for this experiment include 1 piece ripe banana, microscopic glass slide, microscopic cover slip, cutter, laboratory gown, dropper, microscope, gloves, face mask, and iodine solution or methylene blue. For the procedure, scrape up a small amount of the banana flesh. Smear the cells onto a glass slide to make a thin layer. Add 1 drop of methylene blue on the glass slide and specimen. Put the cover slip and make sure that there is no air bubble in the specimen. Slowly place the cover slip on top of the specimen. Put the specimen slide under the microscope and observe. 10 times or low power first, then 40 times or high power next. The ripe banana under the microscope. Our group observed that ripe banana had plant cells that are round to slightly rectangular, thin cell walls surrounding each cell, numerous small dark dots which are starch granules or ameloplasts, cytoplasm filling the inside of the cell, large vacuoles occupying much of the cell. Cells appear closely packed together. Through this laboratory activity, we observed different specimens under the microscope, including onion cells, cheek cells, contaminated canal water, and ripe banana tissue. We learned that plant and animal cells have similarities, such as having a nucleus, cytoplasm, and cell membrane, but they also have differences, such as the presence of a cell wall in plant cells. We also observed that contaminated water contains various microorganisms and that banana cells contain stored food materials. Overall, this activity helped us develop our microscope skills and gave us a better understanding of cell structures and the microscopic organisms found in our environment. Thank you for watching our presentation. We hope you learned something about plant and animal cells and the importance of using a microscope in studying living organisms. Once again, we are Group 2.